Hello boys, good morning. This is meant for class 12th biology boys, and this is for 11th September 2021. As we have been discussing chapter number 6 or 7 in fact, the search for genetic material, that is molecular biology, molecular inheritance in specific. We have been talking in our live classes about various experiments that are conducted by different scientists to prove that DNA is the genetic material. And as part of that, we discussed two experiments. One is the Frederick Griffith experiment, wherein he has used the mice and Streptococcus pneumoniae. And Streptococcus pneumoniae has two strains, as all of you know. One is the S strain, that is smooth strain. It has polysaccharide mucus coat and it causes pneumonia, as you are seeing that orange colored one. Okay, that coat makes them smooth polysaccharide. So since the surface of the polysaccharides is smooth because of the mucus coat presence, it is called S strain. On the other hand, R strain, R means rough strain. It doesn't have any mucus coat, as you are seeing. You can see the spines, rough surface. So it is called rough strain. And out of the two, S is very dangerous because it causes pneumonia, lung disease, whereas R strain doesn't cause pneumonia. And what is that he has done? He has performed an experiment wherein first he has taken the S strain and injected into the mice. Mice is small rat and it dies. Mice dies. And next, step number 2, he has taken R strain, injected into the mice, and the mice is alive. That means S strain is virulent, R strain is non-virulent. Means S strain causes pneumonia, so rat died. R strain doesn't cause pneumonia, rat is alive. Right? Now what he has done, step number 3, he has heat killed the S strain. He has taken the dangerous S strain and boiled it and he has killed. And injected that heat killed S strain into mouse. The mouse is alive because no bacterial cells found in the heart. And now he has gone another step. He has mixed both. What is that he has mixed? S strain heat killed plus R strain. Injected into the mice, and surprisingly mice died. So when the mice died, it is understood that R strain, which is a good fellow, has absorbed something from S strain, who is a bad fellow, and R strain, being good fellow, became bad fellow. So R strain also has become virulent because something has transferred from S strain to R strain. So the mice died. That's why it is called Griffith's transforming principle. So based on the experiment that he has conducted, he concluded that some transforming principle transferred from heat killed S strain to R strain. It enabled R strain to synthesize smooth polysaccharide coat and become virulent. This is due to transfer of genetic material. Of course, he doesn't know that genetic material is DNA. Only he guessed. Griffith guessed that there is something that is transforming R strain to become bad from the S strain, even though it is heat killed. And then to prove this further, 3 more scientists, Oswald Avery, Colin MacLeod, Maclean McCarthy, worked to determine the biochemical nature of transforming principle in Griffith's experiment. What is that they have done? They purified biochemicals, proteins, DNA, RNA from heat-killed S cells using suitable enzymes. Like you see, first they used lipases, removed lipids from the heat-killed S cells. Then carbohydrates are removed. Then proteases, so proteins are removed. RNAs, so no RNA. And RNA is dissolved. DNA also dissolved. So after dissolving everything, what they have done now? They have performed further. Digestion of protein and RNA using proteases and RNases did not affect transformation. Look at that. They have taken the lipids out, carbohydrates out, and they focused on protein, RNA, and DNA. They used protein and injected the heat killed S cells and add R cells. The S cells appeared. That means transforming principle is there, right? In spite of protein being digested. Next, they used RNase enzyme, digested RNA, injected that S heat cells and added R cells. Still S cells appeared. That means RNA is not the transforming principle. But when they used enzyme DNase and dissolved the DNA, they could find that when added R cells, there was no S cells at all, no transformation. So this goes without saying that proteins are not the transforming principle, RNA is not the transforming principle, only DNA is the transforming principle. So we said in Griffith's experiment that something transformed from heat killed S cells to R cells. What is that? That transformed DNA. When DNA transformed from heat killed S cells to R cells became S cells, transformed. And this experiment proved beyond doubt that DNA is the transforming principle. And further, experiment number 3, Hershey and Chase grew some bacteriophage viruses on a medium containing radioactive phosphorus P32, and some others on medium containing radioactive sulfur S35. Viruses grown in P32 phosphorus got radioactive DNA because only DNA contains phosphorus. Already we discussed. DNA is made up of 3 molecules: sugar, phosphate, nitrogenous bases. So since we are using the P32 medium, naturally DNA becomes radioactive. Viruses grown in S35 got radioactive protein because protein contains sulfur. Now, see, medium containing bacteriophage having protein labeled with S32, sulfur 35, S35. Now look at that, only the protein coat is radioactive, whereas medium containing bacteriophage having DNA labeled with P32, look at that, DNA has become radioactive. And in both the pictures you can see radioactive sulfur 35 is seen in the capsule, protein capsule, whereas DNA is getting radioactive because of the presence of P32 in the head of the bacteriophage, because head contains the nucleus and inside the nucleus DNA will be there. This experiment is done by 2 scientists, these 2 gentlemen, Hershey and Chase. It's called blender experiment because blending is done by them. These preparations were used separately to infect E. coli, Escherichia coli. E. coli is a good bacteria. It is present in our stomach and intestine. So after infection, the E. coli cells were gently agitated in a blender to remove the virus particles from the bacteria. See, they infected this. Remember, you must differentiate between bacteriophage and bacteria. Bacteriophage is a virus, okay, that infects bacteria. And bacteria is getting infected due to bacteriophage. So what they have done, they made the E. coli to get infected with the bacteriophage. And after getting infected, the E. coli cells were gently mixed in a blender, just like in mixie, to remove the virus particles from the bacteria. That is the purpose. Then the culture was centrifuged. You know, there is a centrifugal machine even in our biology lab. We put that to separate lighter virus particles from heavier bacterial cells. So bacterial cells are heavy, as you are seeing here. Blending experiment is done. Now you can see because of the centrifugation, radioactivity phase protein is seen in liquid, and in the bottom radioactive phase in the pellet DNA. Okay, so that's how the protein and portion and the DNA portion is separated using the principle of centrifugation. So, for bacteria infected with viruses having radioactive DNA were radioactive. Look at that. The pellets, radioactivity phase DNA in pellets. And on the other hand, radioactive phosphorus detected in cells plus no radioactivity in supernatant top solution. That means the DNA has passed from virus to bacteria. So bacteria infected with viruses having radioactive proteins were not radioactive. Proteins did not enter the bacteria. So what has entered? It is only DNA. So you can see here the no radioactive sulfur detected in cells plus radioactive S35 is supernatant. You can see the top version. So radioactivity phase protein in liquid and bacterial pet. This experiment of Hershey and Chase proved that DNA is the genetic material. So there is no doubt without doubt. How is it is done? Again, overall in 3 steps it is done. So bacteriophage with sulfur 35 labeled is the protein capsule. You can see that red color of the body leaving the nucleus. Nucleus is in black color. Whereas the another one is the radioactive P32 phosphorus. Only head is shown red color because there is nucleus in the head region, and inside the nucleus there is DNA. DNA is radioactivated with P32, right? And when this is infected to bacteria, first is infection. This round one is the bacteria, and you are seeing the bacteriophages, that is viruses infecting bacteria, that is infection. Second is blending. You see the nuclear material from bacteriophage has entered into bacteria. Here also you can see the nucleus has entered there, right? Third, centrifugation, mixing. No radioactive detected in the cells. S35 is not detected and it is supernatant or above solution. Whereas the nuclear material from bacteriophage has entered into bacteria and we could detect the presence of P32. That means it is DNA which is entering, transforming from bacteriophage to bacteria, not the protein. Hence, the Hershey and Chase proved beyond doubt that it is the DNA that is the genetic material, right? Now, once we have proved that DNA is the genetic material, what are its properties? That we will discuss in next class, live class. Until then, take care.